environmental change early career reps. And I'll pass it off to our other early career reps to introduce themselves.
Hi everyone, my name is Lauren. I'm one of the other EC reps, and I'll pass it off to Sarah.
Hi, I'm Sarah, and I'm the third EC rep. I'm the newest one, so yeah, nice to meet you all.
Awesome. We're really excited today for our next early career webinar series. And just a brief introduction about the Global Environmental Change section. The AGU Global Environmental Change section focuses on large-scale changes across the Earth over decades to centuries. We make up a vibrant community of scholars with more than 11,000 members from 93 countries. And we're strongly committed to promoting inclusion, diversity, and equity in our section. And we're really excited for today's talk in our early career webinar series, featuring the GEC Early Career Award winner, Dr. Kim Novick, and her research group from Indiana University on network-enabled science to anticipate and mitigate climate change impacts on terrestrial ecosystems. And Lauren is just going to do a brief rundown of the talks today.
So in this webinar, the group will describe their ongoing work to synthesize data from a wide range of ecosystem monitoring networks, to pursue previously intractable ecological research questions, and to produce policy-relevant information about the climate mitigation and adaptation capacity of terrestrial systems. So I'll go through and briefly introduce folks. So Kim Novick, the professor and PI of the group, Kim will discuss emerging opportunities to leverage flux tower network data and data from a new global water potential network for informing natural climate solutions and for understanding ecosystem drought responses. We have Michael Benson. Michael's analyzing drought-driven tree mortality in the eastern United States using USDA forest inventory and analysis network data, and he's a PhD student in the group. We also have Daniel Beverly. Daniel's a postdoctoral research scholar and is leading a new initiative to confront the soil water potential information gap in Ameriflux sites. We also have Jing Chang. Jing is developing novel approaches to leverage FluxNet tower data and remote sensing information to understand long-term ecological drought trends. We also have Sander Denham, who's a PhD student whose work blends information from flux tower sites and the PhenoCam network to understand interactions between phenological timing, fluxes, and drought. And finally, we have Chen Wang. Chen is leveraging satellite remote sensing and ground observations to better constrain the climate mitigation and adaptation potential of cover crops in the Corn Belt. So the way the webinar is going to work today, we're going to let the Novick Research Group run the show. We're going to hold questions till the very end of the webinar. You can put those questions in the chat, or at the very end of the webinar in the Q&A session, you can raise your hand to ask a question. And with that, I will pass it off to the Novick Research Group, and I'll ask folks if they don't mind turning their cameras off for the duration of their presentation, just to preserve their spotlight, and then we can turn cameras back on at the end of the presentation. Thanks so much, and take it away.
Great. Thanks so much, Lauren and Christina. Can you guys hear me okay? Maybe a thumbs up. All right, great. And let's make sure the screen share works. I really want to thank you all for the opportunity to do this. We haven't even started, and already our lab has had a lot of fun with this exercise. So great idea for a webinar format. All right, I'm just going to try to minimize things over here so we can all see. Okay, so before we dive in, I want to just sort of give a little background on sort of the driving questions for our lab's work. And what we see here are two figures that I bet many in the audience have seen before. On the left is a figure showing on the top half of the panel, historic greenhouse gas emissions from fossil fuels, and then also land use change. And then on the bottom half of the panel is the fate of those emissions, where the dark blue represents the fraction of emissions, about 25 or 30%, that end up in the oceans each year. And the green blob represents the fraction of emissions, again, about 25 or 30% that ends up being sequestered by terrestrial ecosystems. We can see that a big difference between the green blob and the blue blob is that the green blob is much more variable from one year to the next, because the land carbon sink is very sensitive to interannual fluctuations and meteorological conditions. And then the light blob is what remains in the atmosphere. So already we can see that we owe a great debt to the oceans and terrestrial ecosystems, without which we'd be experiencing climate change at a much faster rate. On the right, you see a figure that shows time on the x-axis going from the past to the future. And on the y-axis is land carbon uptake for the whole globe, where each line represents a different model prediction. And the point of this figure is that up until this point, most models tend to agree that indeed the terrestrial land surface is a strong carbon sink, but as we move forward in time, the models begin to disagree on the magnitude and even the direction of future land carbon uptake. And because this is such a huge part of the global carbon cycle, this injects a lot of uncertainty into our ability to prepare for and adapt to climate change, right? So it's not particularly surprising that the models have such a hard time with this challenge, because the terrestrial carbon cycle is a very complex system, integrating processes occurring over scales as small as subcellular in the case of photosynthesis, up to whole plants, where we need to think about water transport, up to whole ecosystems, where we have to think about not only plants, but also the soil, and then entire landscapes where we have to think about all of these disturbance events. And of course, there's many arrows shown in the figure and not shown, representing climate change feedbacks and interactions. So just an awful lot of complexity baked into this system. Nonetheless, undaunted by the complexity, over decades, globally, we've invested huge amounts of time and energy and money into developing tools to better understand the terrestrial carbon cycle. And chief among them, and what's really our lab's bread and butter, are flux towers and networks of flux towers. Flux towers allow us to measure continuously and directly at the ecosystem scale the net flux of CO2, water, and energy between ecosystems and the atmosphere. So just a really great tool for understanding that variability in those fluxes in time and space. And of course, over the years, we've seen other highly relevant and synergistic networks come online, including the USDA Forest Inventory and Analysis Program, PhenoCam, and SatFluxNet, which are some of the networks with which we work. But we haven't just invested in observational networks. We've invested in big, grand global change experiments. We've invested in what is now just an amazing set of remote sensing platforms capable of detecting not only features of canopy greenness and structure, but also directly ecophysiological function. Of course, models are a crucial tool for being able to see into the future. We've also had to come up with some new ways to think about how we teach the next generation of Earth system scientists to work across all of these different platforms. So one example is the Flux course, which runs every year in Colorado. It's a 2-week course focused on both flux measurement and modeling. But the truth is, despite all of this investment of time and energy, we have not yet solved this challenge. You can see it on the left in 2006, and then again on the right in 2014. And remember, it's a hard challenge. It's a grand challenge, right? And we can't expect to see that uncertainty disappear overnight. Certainly, we should keep trying, right? There's always the hope that we'll sort it out and substantially reduce the uncertainty in Earth system model predictions. But the thing is that even until we get there, even along the way, we are learning an awful lot about the individual pieces of this puzzle. And it turns out that this knowledge is highly relevant for a number of other very important, very pressing research needs. And so what this talk is going to be about today is really taking, these tools that were built to understand terrestrial carbon cycling really at global scales and largely into the future, and applying them to questions in ecophysiology and agri-ecosystem sustainability, drought detection and monitoring, and nature-based climate solutions, which are related questions and related problems, but certainly quite pressing as the impacts of climate change become more relevant and clear. Our lab group does this using tools like network-enabled synthesis, where we can think about synthesizing network data across sites. And here are some examples of how we've been able to do that using Ameriflux and FluxNet data, using the same data variables, but collected at many sites across continents or the globe. Our first mini talk is going to be from PhD candidate Michael Benson, who's going to apply similar approaches, but now using the USDA FIA program database to talk about a novel perspective on tree vulnerability to climate change, focusing specifically on mortality. We can also think about network-enabled synthesis across scales, for example, within a site, where our second talk today is going to be from postdoc Daniel Beverly, who's going to talk about the value of generating and interpreting more information about soil hydraulics, particularly in the context of Ameriflux sites. Our third talk is going to be from Sandra Denham, who I'm proud to say is now Dr. Denham. She just defended her dissertation yesterday, and she's going to again be thinking about integrating across scales within a site, linking phenocam imagery and what it can tell us about species-specific phenology as a tool to better understand ecosystem fluxes. And then, thinking about network synthesis across scales, we're fourth going to hear from Ching Cheng, who is going to talk about how she is blending flux tower data and remote sensing data to develop new approaches for detecting ecological droughts in a greening world. Beginning with the observation that as our world gets greener and leaf area index increases, it can obscure drought effects at the leaf scale. And then finally, we're going to hear from Zhen Wang, who is working on a brand new project for us that will eventually involve flux towers, but they don't exist yet. So you can see them here kind of in yellow, but also using windshield transect survey data on the presence or absence of cover crops across the landscape, together with remote sensing information to understand the usefulness of cover crops, not only for their potential to mitigate climate change through CO2 uptake, but also to provide an adaptation tool in how they affect local energy balance. And just before I hand it over to Michael, I do want to say that nature-based climate solutions is an emerging area of research for our lab. We published a couple of papers over the last year or so discussing ways that we can specifically leverage data from a lot of different networks and across many scales to make sure that we are informing these strategies with the best available science. So you can check out either this paper that came out last year in Global Change Biology or a white paper report we published last fall, and you can feel free to reach out to me to get the link for that, or maybe I'll put it in the chat later. But with that, and without further ado, I'd love to turn it over to Michael, who's going to kick off our first talk. And I also want to just check that the audience is still there. It looks like you are. It's always such a trip when you're just talking into the ether. Okay, Michael, you're up.
All right, you can. All right, so a substantial challenge to assess the consequences of climate change is due to the non-stationary nature of terrestrial ecosystems. So ecosystems are very dynamic, and they're shaped by both natural and synthetic drivers. So for example, in the eastern US, due to a colorful legacy of management and disturbance regimes, we've observed this long-term change in forest demographics where we've seen a decline in oak species on the landscape and a simultaneous increase in mesophytic species such as maples, tulip poplar, and American beech. And historically, the ecosystem function of the eastern US has been shaped by this abundance of oaks on the landscape, such that the conservation or regeneration of oaks has been a central focus of many federal and regional management programs. And these programs are motivated by this perspective that oaks are drought tolerant, which has really been informed by a series of studies demonstrating that oaks tend to maintain growth to a greater degree than their non-oak counterparts when drought occurs. However, it's also clear that oaks also operate with reduced hydraulic safety margins, such that this carbon aggressive strategy comes at a substantial cost of incurring greater hydraulic damage and potentially risk for drought mortality. And so this really complicates our understanding of the benefits that oaks can provide us in a warmer and drought-prone future. Will increasing oak abundance help maintain the carbon sink strength, or will it result in greater forest mortality and potential carbon feedbacks? And so to resolve these uncertainties, we really need to have a more concrete understanding of whether oaks or mesophytes are more likely to die during future droughts. And there's perhaps no database better equipped to answer such questions than from repeat forest inventory databases, which these allow us to compare mortality directly on the landscape to assess physiological drought tolerance. And while leveraging these resources to compare mortality is certainly nothing new, often these analyses are done in a way where it can sometimes be challenging to glean something about physiological drought tolerance. And the reason why is historically we've really focused on comparing abundance of mortality on the landscape, which can provide us with some very pertinent mortality dynamic information. However, it's important to consider that abundance of mortality on the landscape is not always indicative of physiological drought tolerance. In reality, the likelihood of survival on the landscape is due to numerous and complex site and climate interactions, such as where species are growing or the climate legacies that they've experienced. And so, we began to question, these analyses that really just compare abundance of mortality. Perhaps they just more accurately reflect that certain species just tend to more commonly occupy, necessarily less physiological, able to deal with it. So how can we leverage these resources to assess relative physiological drought tolerance instead of just differences in abundance and mortality on the landscape? Well, essentially what we did was just rely on Tobler's first law of geography. So if we consider just a generic forest landscape with two species that occupy relatively dissimilar distributions, instead of comparing mortality between these two species in plots that are spatially separated, we can effectively minimize this geographic bias by only considering mortality between species in plots where these species co-occur. In this way, we know that these species experience identical pedo-environmental conditions, they've experienced identical climate legacies, and therefore they experience more similar physiological consequences when drought occurs. And finally, with this approach, what can this tell us about the mesification of the eastern US? So to do this project, we looked at the 22 most abundant tree species in the four-state region of Missouri, Illinois, Indiana, and Kentucky, and we calculated mortality associated with the severe 2012 Midwestern drought. Essentially, we just relied on some very standard methodologies when calculating mortality from FIA data sets, with the important caveat that we're only considering plots where specific species pairs co-occur. And then finally, we synthesized all this information down to develop a drought tolerance index. And here are the results of our drought tolerance index. These are each of our 22 most abundant species on the x-axis, organized from our more drought tolerant species on the left. These are species that had greater survival than their neighbors during the 2012 drought, to our less drought tolerant species on the right, which are species that experience greater mortality than their neighbors. And while this is a lot to digest all at once, we can just go ahead and cue in on what this can tell us about this mesification question. So first, when we look at the oaks, I think what we see is something relatively surprising, that with the exception of white oak here on the left, the majority of the oaks in our study species were actually on the less drought tolerant side of our index, or experienced greater mortality than their neighbors. Next, when we look at the mesophytes, I think we see something that's also a little surprising, that really they tend to be all over the place. And yes, some of our mesophytes, such as Tulip poplar over here on the right, did die in greater numbers than the oak species in our study area. However, some of our mesophytes, such as our maples over here on the left, were among our most drought tolerant individuals in this study region. And so just to quickly summarize what we learned from this is that, sort of minimizing this geographic bias really adds some unique context to patterns of mortality on the landscape. And with this approach, we see that oaks are relatively vulnerable to drought, and especially when we use our metric as mortality instead of just growth. And finally, in regards to mesification, it's perhaps a little bit more nuanced that, this loss of oak species is not necessarily a uniform shift towards increasing drought vulnerability, but will really be determined by which specific mesophytes are increasing in greater numbers. And with that, thank you so much. I'll hand it over to Daniel.
That's right. Continuing with the theme of drought impacts on trees, we're going to hear from Daniel Beverly, who's going to take us to some of the processes that occur before we might see declines in mortality.
Exactly. Thank you, guys. So I'd like to talk about linking the ecosystem flux dynamics, but thinking more on the soil water potential side. And a lot of this work was done by our technician, Alex Cookshanks, and I just want to make sure she gets her recognition for this hard work. As Michael alluded to, drought and drought responses continue to be a grand challenge for ecosystem processes. And we've talked about a little bit earlier how we have spent significant investments in both infrastructure and money in developing new metrics for understanding water content throughout the different ecosystems, whether that being local weather stations, ecosystem scale flux towers, or even global remote sensing products for measuring soil water content. But one of the major issues is that these platforms typically measure soil water content and not soil water potential, that we know is the primary driver of water movement through microbes, soils, plants, as well as controls controls gross primary productivity and nutrient exchange. Thus, we must rely on soil water retention curves to understand and convert from soil water content to soil water potential to improve our understanding of these processes. Additionally, if we look at three different distinct ecosystems in this figure here, we can see that we have temperate forests on the left, needle leaf forests in the middle, and then semi-arid savannas on the right. And regardless of the ecosystem, the soil water retention curve parameters all explain significant variation in gross primary productivity. But one of the major issues is the conversion between soil water content and soil water potential is the nonlinear pedotransfer functions that are used to convert between the two. The typical pedotransfer functions are typically derived from soil textures, which are highly variable and highly sensitive to different uncertainty parameters. For example, we can see the variation in silty clay versus loamy sands and how much variation across a small domain of the soil moisture spectrum we're looking at. Moreover, if we institute a small or subtle variation in any of these parameters, we increase this variability, as we can see the variance or the uncertainty in the error bars or the shaded regions in these individual curves. This becomes even more problematic when we think about trying to do this at larger scales or larger regions across whether the global or ecosystem perspectives, as we typically rely on these remotely sensed platforms for understanding soil texture. And as we can see, for example, in the Midwest, we see very little variation in all the soils representing silt loam. But we know that that's probably not true. And so one of our primary questions is, can we improve or can we increase our confidence in predicting gross primary productivity using lab-derived soil water retention curves rather than strictly relying on texture-derived pedotransfer functions? And so we're going to look at data from 3 different sites, one in northern Michigan and the one in Indiana here, both temperate forests. And then we'll look at a drastically different ecosystem in a irrigated cropland cornfield in Northern California. Starting in northern Michigan, we have the texture model in orange, and we can see how poorly the soil texture model compares to the lab-derived soil water retention curves, where we have a surface curve, a 10 centimeter curve, and a 50 centimeter curve, and how the variation across the soil moisture gradient. We can see that the soil texture model does not capture the wet end of the soil moisture content, nor does it expand to the more dry end of the drought conditions in the sandy soils. When moving to southern Indiana, we can see that the texture model, again in orange, is much closer to the lab-derived curves at the 10 centimeter and the 0 centimeter and 50 centimeter. But we also must consider that this is in a log transform space, so any variation across this curve is a significant deviation in actual nonlinear or non-log space. Finally, if we look at the relationship between the texture model and our lab-derived curves in the cornfield, we see significantly less vertical variation between the 0, 50, and 10 centimeter curves. But again, the texture model does not capture the wet end of the soil water retention curve, which is highly problematic considering that this system is typically irrigated, leaving estimates of soil water potential probably highly uncertain. And so summarizing these figures, the pedotransfer function models fail to detect variations at soil depth, soil organic matter, or soil moisture across the 3 different ecosystems. But when we try to utilize this information, can we improve our predictions of soil water, or can we predictions of gross primary productivity using these 2 new metrics? So commonly when we think about soil water and soil water status, we can think about threshold analysis versus a linear response. And we commonly see in soil moisture dynamics that fluxes or conductances often exhibit a threshold dynamic. So once we reach a certain soil moisture, there's a drastic decrease in carbon flux or evapotranspiration. So looking just at the northern Michigan site, we have the top panel, we have matrix potential. So now it's negative. But so matrix potential on the x-axis and gross primary productivity on the y-axis. And then the panel below is gross primary productivity and soil water content. And we can see the regression is significantly improved when we use the water matrix potential as opposed to soil water content. as we start to remove that threshold dynamic. Similarly, even more impressive is the response that we saw at our soils in southern Indiana here. Thinking about the threshold, around 20% volumetric water content is essentially eliminated once we convert it back into matrix potential. And so we can confidently improve our predictions of ecosystem fluxes across the landscape.
Okay, thank you, Daniel. We're trying to prevent you guys from having too much whiplash here. I do want to say that this water retention curve generation is actually a service that we're providing to AmeriFlux now in collaboration with Rich Phillips' lab and the AmeriFlux Management Project. So if you're a AmeriFlux site and you're on the listserv, check your email for an invitation to participate in the program. Sticking with the theme of integrating measurements and different networks across scales, we're going to turn it over to Sandra Denham, who's going to talk about linking now phenology information from PhenoCam at Flux Tower sites.
Okay, thanks, Kim. Hello there. We're going to jump right in. So we're shifting gears to thinking about phenology and how different species. This is out of order, Kim. Okay, so we're going to think about spring instead of fall that we just saw here. So if you can imagine phenology being the timing of biological events, when we think about vegetation, we would think about, the onset of leaves, and as leaves come on the this graphic is throwing me off because the leaves are going away, but imagine they're coming back on. And this is the start of transpiration and also of carbon uptake. But then, as the season ends, we would see these begin to fall, and thus the cessation of transpiration and carbon uptake. Now, when we think about phenocams, these are tower-mounted cameras at many flux tower sites. These cameras take digital images of the canopy every half hour or so, all day, every day. And this image is from a late summer in September, where we see a pretty homogenous canopy. This is from southern Indiana, Monroe State Forest, kind of our home flux tower site. And what we can gain from this is we can use the green, blue, and red bands, where we can get a greenness index, where we take the green digital number over the red, blue, and green digital number total. And so we can see the seasonal trend in greenness throughout the years, where we see an increase in the green chromatic coordinate, or that greenness index, in the spring as it gradually decreases throughout the growing season and then into fall. But as you can see here from this drone video, our canopy is actually not homogeneous at all. It's made up of many different species that experience leaf off in the fall at different times. You can see purples and oranges and different color greens there. And if you could imagine, in the springtime, you would also see a different timing of spring. Now, historically, we've obtained species level phenology observations from the ground. So there are opportunities with the National Phenology Network that has an app called Nature's Notebook, where you can just get some ground-based observations on phenophases. And we actually have some permanent sites, transects in the footprint of our tower here, as well as other places. And some data that's shown here, if you look at the right-hand y-axis, these are just going to be some leaf categories. But we're really capturing the seasonal trend in leaf on, and then full canopy cover, and then leaf off at the end of the season across these 3 canopy dominant species in our forest. And we do contribute this to the National Phenology Network. But this doesn't come without its challenges, right? So the photo you see there are 2 undergrads that have been working with our lab out in the forest at the ground, making observations sometimes 30 meters above their head. And in dense canopy forests in particular, it's hard to disentangle which tree's doing what at such a large distance. So we wanted to kind of get at a more objective way to look at the individual species seasonal transitions in leaf emergence and then leaf off after the season. And on the left-hand side, what you're seeing is an image from a March image in 2012, where we just kind of, not arbitrarily, but just kind of drew some circles around leaves that trees that had leaves that had merged by then and trees that had not, to get a kind of rough estimate as if we could apply this greenness index to this region of interest to get that seasonal transition in greenness. And even with this at this kind of like coarse scale, we were able to do this. So we're seeing that one tree canopy is emerging about 11 days sooner than the other. So knowing this and then looking at the photo on the right where we can, the fall colors where we can actually start to really see individual tree canopies, we figured that we would be able to, draw polygons around those individual canopies. And by leveraging we ran a boom lift oftentimes in the summer to get some other leaf level measurements in the canopy. So here we had Daniel, who you just saw speak, helping me out fly some mylar balloons that are brightly colored enough that we could actually see them in the phenocam image down at the hut in real time. So in this way, we know exactly what that species is because we can identify it. And we did this for 16 or 17 individuals across the phenocam image in the foreground there. And we fit the greenness index for those individual regions of interest and tried to compare it with what we were observing on the ground. And we actually do a pretty decent job of capturing when leaves are turning on in the spring and the beginning of the year and when they're coming off at the end. But what we don't see is, that gradual decline in that greenness index as the season progresses throughout the growing season. So we feel pretty confident that we're actually capturing these correct species, and that our phenocam is able to better inform what we're seeing there. So then we can, I use this R package called PhenoPix. This is the package that allows to, draw around individual canopies or any region of interest really, and will allow the extraction of those, that green chromatic coordinate or that greenness index. And then by fitting this double logistic model, we can actually get at the seasonal trend that we see in the green chromatic coordinate and then start to assign some more biologically relevant metrics and phenophases. So on the left, you see the green up, maturity, senescence, and dormancy. And if we run enough iterations of this model to those individual green chromatic coordinate seasonal transitions, we can get more discrete things like the start of spring, peak of photosynthesis and POP, or EOS of indicating end of season. And for my purposes, I've been really more interested in the start of spring and how that can influence interavailability in ecosystem level processes like evapotranspiration or growth primary productivity. So when we take, our camera has been up since about 2009. So when we take 13 years of data across 16 individual canopies that span, there's actually 5 species that we looked at, but the ones on the bottom are actually the 3 that had replication in our viewshed. But what's notable here is at the top in panel A, we're actually seeing, there's the variability within the year across these individuals. We're seeing some years where, leaves are really coming out pretty much at the same time. And in other years where there's a lot more variability. On the bottom, we're seeing the variability across those 13 years within these 3 species of sugar maple on the left, tulip poplar in the 3rd position, and sassafras on the far right. And just kind of looking at how that compares to the full view, which was also a region of interest that was drawn around the area encompassing each individual species. Importantly here, we see that tulip poplars are quite variable across our canopy and across these years. And the probability of leaf emergence at certain times of the year are kind of variable, where you look at something like the sugar maple, where the start of spring date of year is the probability of it occurring around the same time is pretty equal. And then if we take a look at the average day of year that we would expect our forest to leaf out across the entire canopy from that full view shed, and then look at the variability within and across these species, we really see that our tulip poplar is leafing out consistently sooner than the rest of our canopy dominant species. Moving forward, again, this is largely a methodological approach for some of the newest work that I've been interested in. But moving forward, it would be this was what I'd shown was just from Southern Indiana at Monroe State Forest, is to expand to other sites, other eastern deciduous sites that have mixed species, to look at how that variability exists across different forests in the eastern US, and link that to higher level ecosystem processes, and leverage the flux tower meteorological data to really get into species level sensitivities to different meteorological drivers.
Okay, great. Thank you, Sander. Sorry, I probably accidentally deleted your leaf on slide when I compiled everything together. Next, keeping with the theme of greening and drought, we're going to hear from Ching Cheng. Who's going to talk about ecological drought monitoring.
Thanks, Kim. So today, I'd like to present how can we detect ecological drought and its changes in a greening world. I'd like to start with the evaporative stress index. It is ESI. So it is defined as the standardized anomaly in the ratio of actual ET to potential ET. So ESI has been known as a better proxy for ecological drought detection than meteorological drought indices and hydrological indices. This is because ESI considers decoupling of ET and PET. So ESI is able to reflect the quickly response of stormwater conductance and the dynamic response of plant to soil moisture decline and atmospheric moisture decline during drought. However, greening is affecting the ESI's performance. To present the reason, I'd like to use an earlier spring year. So earlier spring year is supposed to have a higher RAI and a higher ET than normal years during spring and early summer. That is greener spring and greener early summer. So if we calculate ESI, we got positive values, but the positive value reflects no ecological drought. If we want to see more ecological drought information and greening, we have to remove the effects of greening. So a possible way is to do a RAI correction for the ESI. So we develop a RAI corrected ESI by using a RAI related PET. So in the equation, the PET is the expected maximum ET during well-watered conditions that can reflect the changes in RAI. So how does the corrected ESI work for ecological drought detection? Let's imagine there's a drought during spring and early summer for the earlier spring year. So the normal ESI cannot see the drought. This is because the annual changes and annual increase in ET due to the early default could be more than a limitation to ET from drought. So ESI is positive. However, the area corrected ESI can see the drought. This is because the area corrected ESI has considered the changes in RAI, and also it can help us to detect the annual changes in ET linked to physiological changes or physiological limitation from drought. So we did a test in Monroe State Forest, and we selected the year of 2012. This is because the year has been identified as an earlier spring year. From the modest RAI, we can see the RAI in 2012 just increased earlier and much higher in spring and early summer than normal years. As for the ESI calculated for spring and early summer, marked in gray shaded area, we can see the original ESI are positive, which means the area which means the original ESI cannot help us to see the ecological drought. However, the area corrected ESI are much lower than the original ESI, and also they are mostly in negative values, which means the area corrected ESI help us to detect the ecological early drought stress. So how's the changes of ecological drought in long-term and global scale? We did analysis by using FluxNet Flux Tower network, and we found that by using normal ESI, there's no changes by using normal ESI, there's no changes of ESI along with the increase of RAI. So the normal ESI did not see ecological drought changes along with greening world. However, when we are using RAI corrected ESI, we can see that the RAI corrected ESI decreases significantly along with the increase of RAI. So the RAI corrected ESI just reveals the increase of ecological drought along with the greening world. That's all I have. Thank you.
Thank you, Ching. And we're just about finished. What I like about this exercise, it's fun to see how the evolution of our lab's focus has changed over the years. And we're sort of going in reverse order of when these folks arrived in the lab. So our last speaker is working on one of the areas, which is a new focus for us. It will be Shen Wang talking about cover crops as a natural climate solution.
Thank you. We have been discussed about water, energy, and carbon cycles. So just based on all these understandings, we're trying to better manage our ecosystem. So here we focus on carbon belt region. We specifically focus on cover crops. So the cover crops is the vegetation we implement during the off-season, usually from the October or November to April or May. So when we put the vegetation in this ecosystem, we actually changing their water, carbon, or the energy balances. Specifically, we're thinking those vegetation can take the CO2 from the atmosphere, but also can change in the local water cycle and increasing the water and soil quality, but also it might have cooling effects. In long term, we're thinking those benefits can buffer the extremes and improve the cropland vulnerability and potentially increasing the crop yield. However, those benefits are actually based on the limited point data resources. So what we really want to understand is all these benefits will be applied to the entire corn belt region because we want to implement cover crops in this region. So what we have is a great ground truth data. So is professional people drive the car across our state and to give us the information about the field with the cover crops or non-cover crops. So for each field, we get remotely sensed data. So specifically, we focus on carbon sequestration potential proxies. So we have these three indices here. We also have land surface temperature to indicating the cooling effects. Just show some initial results for Indiana. So we focus on the southern Indiana counties here. So we have these three indices to indicating carbon sequestration potential. So the green color indicating the cover crops, and then brown color indicating non-cover crops. And then x-axis is October of this year to the October of previous year to October of this year. So during the off season, you will see the cover crops has larger carbon sequestration potential, and which is consistent for the three indices. When you go to the growing season, you see the carbon sequestration potential actually is shifting because we did something during the off season, right? We also calculate the temperature difference here between the cover crops and non-cover crops. So the negative values will be the cooling effects, and then the positive values will be meaning the warming effects. Still, the off season, you see more cooling effects, but then the growing season, you see the warming effects. But actually, when you're thinking the whole year, the warming effects and cooling effects actually compensate in some way. So it might not provide actually cooling effects for the cover crops. We also find the location are pretty important. So here we highlight the Decatur County, which is also in southern Indiana. We're also showing the time series of carbon sequestration potential for cover crops and non-cover crops. The time series is pretty similar to the counties we described before. But when you go to the middle Indiana, the patterns is already changed. When you go to further northern part of Indiana, the two counties does not have difference actually for the carbon sequestration potential. So our initial conclusions is that the northern part of Indiana, the cover crops probably have carbon sequestration potential, but may not have cooling effects. But we really haven't really thinking about respiration or albedo change. So our lab actually trying to install a couple at covariance flux tower sites for the cover crop project, to thinking about net carbon flux instead of carbon sequestration potential. But also myself now exploring a lot about albedo, just thinking about if we put the cover crops or leave more residues on the land compared to the bare ground, we will have more light to reflect back to the atmosphere, right? So that is also a cooling effect. But I think the most important thing for the cover crops project is that we need to find the right locations on time, even the species for the cover crops in the corn belt, so that we can maximize the climate benefits of the cover crops.
Great. All right, and I'll just wind us down here by saying again, I really appreciate the opportunity to do a webinar in this format. It was a challenge to try to think about how the different pieces of the lab, which you can see are somewhat disparate, fit together. But again, I think that it amounts to leveraging this decades of research infrastructure investment for tools in monitoring and forecasting land carbon cycling, and applying some of those tools to some really interesting questions regarding ecophysiological mechanisms, agroecosystem sustainability, drought detection and monitoring, and natural climate solutions. And I do want to, before we leave, I want to mention, the title of this talk had something like network-enabled science or network-enabled synthesis in it. And that's a big part of what we do. And so we're really indebted to all of the people out there that have shared their data to environmental observation networks. And hoping to pay it forward, I'm excited to announce that we're kicking off a new NSF-funded research coordination network very soon. It's called SciNet, and you can see my fabulous team of collaborators here. But our goal is to fill that gap in water potential information so we can better connect environmental drivers to ecosystem responses. So we'll be creating an open database of soil and plant water potential time series from across the globe, developing best practices for water potential measurement, including continuous approaches, and providing a wide variety of early career training workshops, networking activities, and working to build a more diverse and inclusive community. So with that, I do want to acknowledge many of our lab members who didn't participate today, in particular, technicians Mike Voiles, Alex Crookshanks, and Steve Scott, without whom we wouldn't really do anything. As Steve, you can see here, is our resident daredevil. There he is, and there are his feet as he's always paragliding around, taking fabulous pictures of our tower from above. I'm grateful to work with so many people who are willing to go to such great heights and great depths to understand land-atmosphere interactions. And you would think I could have stopped there, but I didn't, or that maybe I would stop at this slide. But no, it's it's really fun to have such an engaged and creative group of young scientists to work with. So now we really are finished. And if there is time for questions, we'll be happy to take some. So thank you all very much. Now I'm going to try to get the gallery view back so I can see you again.
Thank you so much. That was so great. And thank you also for sharing all of the pictures. I always love as an added addition to group presentations. So if anyone has questions, like we mentioned before, feel free to raise your hand or to put those questions in the chat. And so I see we have one hand raised already. So we'll go ahead and you can go ahead and unmute.
Hi, everyone. First of all, great talk. I really enjoyed it. Thank you very much for that. So I have a question for Daniel, where he says the improvement in the soil water potential that improves the estimates of GPP. So my question is, how did you make sure the improvement is only because of the improvement in soil water potential? Because GPP may be controlled by many other factors, including leaf water potential, and there are many other parameters. So how did you make sure the improvement is happening only because of the soil water potential?
So my interpretation of the question is you're asking how did I improve prediction of GPP? It's just improved relative to soil water content. And so is that?
I think, yeah, I'm not sure if you're talking about the results at the end where you showed the improved correlation.
Yeah.
Yeah.
Is that the question? Or
yeah, I mean, how you're saying the improvement is only because of the improvement in the soil water potential, not because GPP, how you're defining GPP, like how you're estimating GPP, because there are many other factors playing roles in the estimation of GPP, right? So how?
Sure.
Like
so
yeah,
I think the easiest way to think about this is it's all the same data. The GPP was measured from the flux tower, or modeled from the flux tower. And so that site-specific soil water content was converted using that soil water retention curve, and it just improved the correlation. It converted, made a more linear response rather than a threshold response.
All right, thank you. Yeah, that makes sense to me.
Thank you. I see we have another hand up, Matthew.
Yeah, hello everybody. Thanks you all for a great talk. I have a question for Xian. You highlighted some attempts to look at the cover crop impacts using more of the carbon uptake, or carbon sequestration, like through photosynthesis, greenness metrics. Are there any approaches to look at the respiration side of how those differences could tease out using remote sensing?
I don't know, sorry. I don't think, I mean, from my knowledge, I don't know we have respiration proxies for remote sensing. That's why our group trying to build on the add covariance for the cover crop project, so we can incorporate respiration for the cover crops. But I don't know anything, I mean, personally.
Yeah. Okay. Well, thanks.
Yeah, thank you.
I think it would be interesting to look more into that. But thank you.
Yes, thank you.
Yes. You can sort out how to estimate NEE or RE from space map. That'd be great. Just
oh yeah, that would be excellent.
So it looks like we have a couple things in the chat. There was one comment from Michael that says the result about cover crops not producing a cooling effect is interesting and seems like an energy balance with albedo and actual evapotranspiration might be useful. Reminds me of some past work by Morton on AET. And then we have another question that says, how do you go from eddy covariance measurements to GPP?
all right, I'll take both actually very quickly. I absolutely agree with the question about the cover crops and energy balance. You know, when Shen was presenting it, looking at a sort of a local cooling effect, which can be very important, particularly for farmers, right, where temperature matters a lot when they make their decisions to plant and that sort of thing. Albedo is a little different than temperature because it can you know, just reflect shortwave radiation back into space where it would not have the opportunity to stay in our atmosphere and heat our surface. And so we're really looking forward to diving into those questions as Shen continues her regional scale work, but also once we get those flux towers installed, because they measure all components of the energy budget. As for getting from GPP or eddy covariance to GPP, I'm afraid I cannot answer that succinctly, but it relies on models and you have to make some assumptions. And if you really want to know, I can recommend you consider applying to next year's flux course, where that question is answered over the course of about a day and a half. But it is important to remember that flux tower derived GPP is not a measurement. It is an estimate derived from a model strongly informed by measurements, but not a direct measurement.
Awesome. Thank you so much. I think we have 3 more questions in the chat, and that might take us to our time. So there's one from Tyson that says, question for Daniel, do you have any hypotheses as to why the matrix potential is able to explain more variance at one site over the other? And I'll pause and we can go ahead and address that one.
Yeah, so especially the 2 deciduous hardwood species, the 2 deciduous hardwood forests. Northern Michigan is very, very sandy soil and almost a homogenous texture throughout, while the soils for Morgan Monroe or Indiana here had much better both density and texture variation as we went in depth. And so I think that the texture and the siltiness and the clay-rich soils here improve water retention and drive most of that variation.
Great. And we have our next question. Great talks. Did you consider any soil data in the last project? Does soil texture / chemistry change significantly as you travel north to south across the state? And then I'll wrap that in with the last question, which is a question about cover crops related work. Are you all considering doing these measurements for senesced crops not cleared, or even senesced / unburnt grasslands during winters? Any thoughts on albedo cooling effects of such covers? So I think, yeah, with those last two, we'll take it away.
Yeah, I think Shen should answer those with the note that not senesced could also be interpreted as no till.
Yes.
Okay.
We have been thinking about tillage. Tillage practice or no tillage practice so that it can have more like residues on the land, and those residues actually reflect a lot of, we assuming reflect a lot of light back to the atmosphere, which is also kind of albedo effect, which is a kind of a cooling effects we are talking about here. I think the soil data, I think we're also thinking about the soil texture and soil color, since we have maps that I was looking at Indiana. It doesn't have a, I mean, from my eyes, I mean, probably have big difference, but from my eyes, I didn't see big difference from the soil color at this sense. So I think we're more thinking about probably snow impact instead of soil color impact at the current stage. But if we find more like soil data on the ground, we might go further for that. But I don't have soil data from field right now. But I think it's a really interesting topic to go forward. Yeah. Am I answer all the questions? Okay. Well.
That was great. Well, thank you so much. I think we are right about to hit 2 o'clock. Before we leave, I just want to give another round of applause to the Novick research group, and thank you all so much for being here. And that we in the GEC section really appreciate you all being here, and we're happy to highlight the work of some of these early career researchers. So keep an eye out for some of our future webinars. We hope to continue this series featuring other GEC early career winners. And with that, I want to thank you all again for being here, and we will see you next time. Take care.
Thank you. Thank you guys so much for hosting. That was so much fun.
Thank you. It was fun watching. We appreciate it.
Yeah.